Hello, and thank you for joining the Food Safety Educational Conference as part of the 2021 virtual Southeast Regional Fruit and Vegetable Conference. I'm Billy Mitchell, the FSMA Training Coordinator with the National Farmers Union, and I'll be the moderator for this presentation. I'd like to thank Highland Ag Solutions for sponsoring the 2021 Food Safety Educational Conference. Let's watch this short video provided by Highland Ag Solutions.
Highland is virtualizing the business of agriculture. From soil to sale, we are uniting growers, packers, marketers, and their customers in ways never done before. In today's global marketplace, competition has never been fiercer. You need innovative tools to provide business and market transparency for the industry to remain sustainable for future generations.
Pesticide CEU and CCA credits are available for most presentations offered during the live Southeast Regional Conference. Check the Pesticide CEU guide for a list of approved presentations and participating states. The Pesticide CEU guide is located in the Event Resources tab under the Media Player. The guide is also available at the Resources Center located on the main menu of the conference platform. Please note, pesticide credits will only be available for 1, registered attendees, and 2, only during the live conference. Credits will not be available for on-demand viewing. There's a simple 3-step process to receive pesticide CEU and CCA credits. First, go to the audience chat box located on the left side of your screen and type your first name, last name, and the states for which you are requesting credits. Again, go to the audience chat box located on the left side of your screen and type your first name, last name, and the states for which you are requesting credits. You will need to do this for every presentation to request CEU credits. Second, the second step is to sign out at the end of every presentation. To sign out, go to the audience chat box and type your first name, last name, and the states for which you are requesting credits. You'll be reminded to sign out at the end of the presentation. The third step is to complete the pesticide CEU registration. You only have to complete this registration one time during the conference. This is not required with every presentation. To access the pesticide CEU registration web link, open the pesticide CEU guide on the Event Resources tab located under the Media Player. The pesticide CEU registration web link is located on the front cover of the guide. This presentation is pre-recorded to reduce technical difficulties. We will be answering your questions live at the end of the presentation. You can submit a question at any point during this presentation by typing your question in the questions box and pressing send. Don't forget to thank our 2021 conference sponsors and exhibitors by visiting the virtual trade show and featured products pavilion. Lastly, don't forget to join us each morning for coffee chats and each evening for networking. Check the conference agenda for details. Now, I'd like to introduce Dr. Ortega, Associate Professor at the Center for Food Safety with the University of Georgia, who will be presenting What is Cyclospora? Dr. Ortega is an associate professor at the Center for Food Safety, and her research is focused on food and waterborne pathogens. She is currently studying the dynamics of disease transmission that take place in the production and processing of food products. She has authored peer-reviewed manuscripts and book chapters, and is the editor of 3 books on foodborne parasites. This topic is important because in the US, we had recent foodborne outbreaks associated with Cyclospora, and in 2018, and 2020, the implicated products were grown and processed in the US. And now I'll hand it over to you, Dr. Ortega. Thank you.
Thank you. Good morning. Again, I've been working with Cytospora for a few years, and I'm glad to be at this meeting to tell you a little about what Cyclospora is. Cyclospora, as we know, we have several types of foodborne outbreaks. We have viruses such as hepatitis and norovirus, bacteria such as Salmonella and E. coli, as well as parasites, flagellates, ciliates, amoeba, and coccidia. Cyclospora cayetanensis is a coccidian parasite. But one of the uniqueness about this group of parasites is that it's a eukaryote. Bacteria are prokaryotes, so it adds a big, it has so many different characteristics compared to the bacteria as well as the virus. Let's start with what causes Cyclospora infection with Cyclospora. Again, it's a coccidian parasite that will cause diarrhea, nausea, fever, vomiting, anorexia, and weight loss in individuals that acquire this infection. This parasite cannot be grown in media like what we do with bacteria, so there's no enrichment media, and it requires a host to be able to multiply. So an individual that has a cyclosporiasis, that's the name of the disease of Cyclospora, basically, if we look in the stool samples of these individuals, you will see this type of structure, which is the unsporulated oocyst. This is usually the diagnostic stage of the parasite in humans. However, if we collect these samples and we store them for 7 to 15 days, you will see how these oocysts change in shape. Now you will see that these oocysts will have two different shapes, and each one of these ones have two little worm look-like organisms, and they are called sporozoites. Again, let's remember this. This is very, very critical. Again, if we look at stool samples of an infected individual, you will see that the oocysts are basically round spheres with amorphous mass inside. That's what we call the morula. And again, 14 to 15 days, these oocysts will differentiate and they will form these two sporocysts each one. One of the characteristics of this parasite is that if we look at them using a microscope that has UV light, these oocysts will autofluoresce, which means they're going to be glowing light blue or light green, depending on what type of filter we use in the microscope. But basically what it means is that the UV light is coming into the parasite and it's gonna be bounced back and captured by the microscope. What is interesting about this parasite is that if we freeze it or we expose it to very high temperatures, the parasite will die. Again, it's a eukaryote. We can filter because this parasite measures about 8 to 10 microns in diameter, so we can filter. But we cannot inactivate it. It's a highly resistant microorganism. And there's a lot of questions whether we can kill this organism by some type of radiation. One of the reasons why this organism is highly resistant to chemicals is because of the cell wall of this organism. You can see here it has a double cell wall. It protects the sporocysts and then the sporozoite. Here's the sporozoite. This is the nuclei of the parasite. So any chemical that is going to be affecting and killing this organism first needs to break this barrier, the second barrier, and then the third barrier. The good news is we have a treatment for this parasite. Trimethoprim, sulfamethoxazole, is commonly used around the world, and it's very effective at controlling this parasitic infection. There's also been some tests and some publications on the use of ciprofloxacin. We all call it ciprofloxacin. It's very effective, but there's been some conflicting publications again. Some groups say that it doesn't work, that it can be a second alternative for trimethoprim sulfamethoxazole. This drug, the common name is called Bactrim. Now, if we look at what's going on, in particular in the US, this parasite was described in the early 1990s, and we have been having outbreaks throughout the years. You can see here 1996, 97, all the way down to 2000. And if you look at here, the implicated food products having berries, having herbs, fresh basil, or salad mixtures. All of these products are usually consumed raw. So this parasite has not been unusual to be found here in the U.S., but most of these outbreaks have been associated with products that have been imported from another locations worldwide. It hasn't been until 2013 that we started to see more cases of Cyclospora in the U.S. We had a very large outbreak actually in 2013, and you can see here there's two peaks. The very first one occurred in June, and the second one occurred at the end of June and the first of week of July. This large outbreak caused 643 illnesses in 25 states. And again, the very first peak the highest number of cases occurred in the states of Iowa and Nebraska. And the second one was in Texas. The first one was associated to a salad mix that was obtained from or was imported from Mexico. And the one from Texas was associated with cilantro that was also imported from Mexico. The next year, again, we had 304 cases in 19 states. 64% of the affected individuals were from Texas. And all of these cases were related to consumption of cilantro that was imported from Puebla, Mexico. In 2015, pretty much the same situation. We had 546 cases. And again, all of these outbreaks that were occurring 2014, 2015, and so on, were occurring during these months of the year. Again, in 2015, the culprit was attributed to be cilantro, again imported from Mexico. 2016, we had 384 cases, but the source was not identified. In 2017, we had, there was a big change here, because now in 2017, we were looking at all the cases that were occurring in the US. We were not just looking at the outbreaks that we could identify the source of the outbreak. So we knew that there were 1,065 cases, of which 597 were associated to an outbreak. In 2018, things got very, very serious in the US because we had two outbreaks. Here you can see the epi curve. The first one implicated broccoli, cauliflower, carrots, and dill dip. These are the blue bars, and again, you can see here occurs in May, and it ended sometime in August. The second outbreak occurred, it's the yellow bars, and it was associated to a salad mix. The first outbreak, the one with broccoli and cauliflower, were basically trays of these vegetables that were sold in gas stations and certain stores, whereas the other one was associated with salad mixes that were sold and distributed by McDonald's, actually, and in the restaurants. And the provider of these salad mixes, the processor, was located in Stringwood, Illinois, and it was from Fresh Express. One of the challenges that we saw in this case was that there was an investigation on where these vegetables were grown, and it was pointed that it was in California. A few samples from some of the fields, not related to the outbreak itself, but from the same location, tested positive for Cytospora. So this was the first year where we actually had evidence that Cyclospora was actually contaminating product that was produced and processed in the U.S. In 2019, again, we had other cases of Cyclospora. Now we had a new scenario, a new situation that was going on in the clinical setting. There was culture-independent testing done, which meant that when a patient came with diarrhea. In this case, the stool samples were tested for a number of pathogens. And by molecular testing, it was determined that 2,408 cases of Cytospora were detected in this year in 37 states. Again, this is very important because the numbers are higher, and to a point, this might be related to that use of new diagnostic tools in the clinical setting. What is also interesting is Cytospora has always been considered a disease of travelers. In this case, these 2,400 cases were individuals who did not travel 15 days before illness. And why do we say 15 days? That's about the time a person will develop symptoms since ingesting the oocyst. So again, all of these cases would be considered domestic cases, not from travel. Again, in this during this year, we had multiple clusters of infection, but there was one that could be clustered and basically affected 241 individuals with in 15 states. And the commonality on all of these cases was fresh basil that was imported from Morelos, Mexico. Something very important also happened that year. When there's an outbreak, CDC produces a questionnaire in order to determine what are the implicated products that could be responsible for an outbreak. In this case, this questionnaire, which is called the Cytospora National Hypothesis Generating Questionnaire, was updated and went virtual, which meant that a lot of the health departments in the U.S. were able to enter data much faster than before. There was everything done on paper. Once again this year, we had 1,200 cases of cyclosporiasis in the U.S. And again, these were from individuals that did not travel 14 days before onset of illness. As of September 24th, we had 701 cases. 14 states were affected, and there were 38 hospitalizations as a result of this outbreak. If you can see here, the states that were mostly affected were in the central states of the U.S., and the implicated product, again, was a salad mix that was produced by Fresh Express, also that was processed at a facility in Streamwood, Illinois. So the consequence of that, of course, was trying to do that traceback investigation, trying to determine where is it that these lettuce and cabbage and carrots were produced. The red cabbage led the investigation from all of this outbreak down to Florida and South Florida, actually. And FDA tested water from the canals, the water canals, and they tested positive for Cytospora. So the big question now is what's going on in the U.S.? Do we have Cytospora? Are we endemic for Cyclospora, which basically means that the parasite is resident in the United States? Or because, again, we know that when we import products that are contaminated and they're coming from endemic location, yes, we're going to have outbreaks here in the U.S. But this basically, that the outbreak of 2018 and 2020 makes us rethink this theory that perhaps Cyclospora is already resident in the United States. So what happens in endemic location? I mentioned to you that we had several outbreaks and they were associated with imported raspberries, imported herbs, and imported salad greens. So again, the very first outbreak that occurred in the U.S. was associated with consumption of raspberries that were coming from Guatemala. So Byrne and team did this study in Guatemala, and this actually repeats in every single endemic location of, where Cytospora is present. Okay. So basically, we can see that children are the ones that acquire the infection. and there may be or may not be diarrhea. That's demonstrated. That's also backs up the idea that perhaps the first infection, the person will develop severe diarrhea, but with repeated infection, the person is developing an immune response. And of course, the illness is going to be shorter and perhaps asymptomatic, which means no symptoms. The other interesting situation with Guatemala was that the cases of Cyclospora were detected during the months of April all the way down to August, September. Remember when I show you the tables of the outbreaks or the graphs of the outbreaks that happened in the US? Those happened exactly during those months. So there was an agreement that what was going on in Guatemala and when was the high season of Cyclospora in Guatemala was also happening here in the US. And seasonality is not unusual for Cyclospora. If you look here, for example, in Nepal, many years ago, before it was actually described as Cytospora cayetanensis, it was called CLB. And CLB stands for cyanobacteria-like bodies or coccidia-like bodies. But what you can see here is that in Nepal, it happened in expatriates, people that were going to hike the Everest. They will come out with gastrointestinal illness. And this would happen during the months of July, August, September, and it will repeat every single year. Something similar happens in Mexico. This is a study from 2000 to 2009, and we actually have another paper that will be coming up soon. It is from 2010 to 2019, and pretty much has the same peaks. The seasonality is very marked and occurs during the months of April to August. Every single year it repeats. Now, something very similar happens in Peru. In Haiti, for example. Both of these countries have their peaks, their seasonality, but occurs during different months of the year. It occurs during the months of January, February, March, and April. Every single year it repeats. So we know that our seasonality in the US coincides with what goes on in Mexico, with what goes on in Nepal. It does not coincide with what happens in Haiti as well as in Peru. So now we know that Cytospora has two very unique commonalities. One of them is the seasonality, and the other one is that this parasite cannot multiply in the environment, and it requires 7 to 15 days for it to become infectious. That is the only parasite in humans that does that, that requires that time to mature in the environment. So what goes on in the environment? That's something we're trying to figure out. There is one way could be the water. If we have contaminated water that is used to irrigate crops, agricultural water, which can be during pre and post harvest, or perhaps that soil is contaminated, and at some point the vegetables also get contaminated. In the case of cilantro, for example, there is not much processing going on. Basically, the bunches of cilantro are collected in the field, and they may be rinsed or not, depending on how busy the season is. And they get placed in containers with ice, and they get shipped into the US. So there really isn't much of a control point during the production of cilantro or basil, for that matter. Now, as I mentioned to you, water is rather critical for not necessarily for propagation of the organism, but for dissemination of the organism. We have looked in Peru, we have looked in Mexico, and we know that during the high season of this parasite in the human population, we can also detect it in the environment. We also know that other studies have done have looked for Cyclospora and Cryptosporidium, and definitely if there is a town, there's a potential of contamination of surface waters by humans, and therefore there will be more positives downstream than upstream of the town. Now, let's remember that Cytospora is exclusively anthromorphic. What does that mean? That means that this is at least until now, we think that Cytospora can only infect humans. So we cannot say that an animal was responsible for contaminating the water. The contamination is going to occur with human fecal samples. Okay, so again, everything that is happening now, it was not new for some of us because in 2013, a Canadian study basically purchased bad salad greens that were produced in the US, in Canada, and in Mexico. And what they looked for the Cytospora. Cryptosporidium is another coccidian parasite, and Giardia is a flagellate. These three are parasites. What was interesting is they detected all of these three organisms in salad greens. And if you can see here, most of the bags that were tested were from the US. So again, one of the challenges that we have working with parasites is that if we do molecular testing, we're determining the presence of DNA of these organisms in the leafy greens, in the salads or any product that could be present there. But we cannot say whether they are alive or dead. All of these cases that were positive were not associated with any outbreak. That is very different from bacteria, for example. If you find a positive, it's because the bacteria was able to grow and multiply in a specific medium. The other challenge that we have again with Cyclospora is that this parasite is actually can do very well at room temperature and refrigeration. Again, that this organism will be able to sporulate very well at 23 degrees. At 4 degrees will eventually sporulate. It will take longer, but it will sporulate. How do we kill it? Again, one way is to freeze it. If we freeze it, we will kill it. Or if we expose it to high temperatures, we also will be able to kill the parasite. There's been several studies on other alternatives, and that's something that we, even in my lab, we're looking for, trying to see how we can kill this organism, in particular with fresh produce, because again, we're talking about cilantro, we're talking about basil or salad mixes. None of those can be frozen or heated at these type of temperatures. That will kill for these periods of time. That will basically damage the product. So at this point, we don't have a particular treatment that will be good to inactivate this parasite, especially in products that are consumed raw. We also know that if the water that is used for applications of pesticides, these chemicals will not kill the organism. The parasite will remain viable. And again, at different concentrations and treatment times. What is interesting, though, I mentioned to you that the oocysts autofluoresce. That means that they are light blue when they are exposed to UV light. That allows this organism to remain viable and be resistant to the solar radiation. And again, this is by looking at how this organism behaves when we change the temperature relative humidity in UV light. So you can see, again, this parasite will remain pretty much all detectable throughout that time. We stopped at day 23, but probably will go something like that. But whereas if we were to look at the conditions, more protected conditions, I should say, again, this would be for more protected conditions. But if we look at where there's actual sun, there's rain, there's cold, different types of temperatures, the presence of the oocysts will basically be reduced. This is something that is, the prior study that I showed it to you comes because of Dr. Robert Sonch did a study on the Giardia and Cryptosporidium. Again, I mentioned to you, Giardia is a flagellate, whereas Cryptosporidium is a coccidian. And these oocysts can survive for longer periods of time. And so they could survive up to 9 days, again, if they're kept at cold temperatures. And all of these, as you can see, they have, we can call it protected conditions because they're inside the house. If they were to be in the fields, of course, these organisms will not last as long. And we actually have done some studies with Cryptosporidium with that. And definitely they don't last very long. And again, because it has to do with the relative humidity, temperatures, and UV rays. Cryptosporidium does not have that protection that Cytospora has with cell wall. So how does this parasite come into to contaminate and get into the crops? One of them is if we use agricultural water that is contaminated, if this is again rivers or it can be canals. There are surface waters that may make it to the fields, to the crops, get in touch with the crops or may not. But regardless of that, if we detect the parasite in these waters, that gives us an idea that potentially they could get into the crops. There's a possibility also that the soil gets contaminated or the harvesting tools, the knives and any tool that is in use, containers, etc., that are used during the harvesting could be responsible for the introduction of this parasite in the crops. Field workers, same thing. If they are sick, they don't have the proper hygiene and they don't wash their hands properly, they could contaminate the field, so they could contaminate the crops that they are. Again, hands, shoes, and clothes could be part of a source of contamination of these crops. During post-harvest, we can have water that is used again for rinsing of these products. And again, if a person that is working in the processing facility and is ill can be responsible for introducing the oocysts with their hands that are contaminated or in their clothes and being in contact with a particular crop. So what has FDA done during these past years? They have developed a tool to detect, it's a qPCR to detect the presence or absence of Cyclospora in products. And you can tell it's a little elaborate, but it has been used during the past years to identify what are the sources of contamination. Now, they also have developed a method for detection of Cytospora in agricultural water. The detection limit is 6 oocysts in 10 liters of agricultural water. Again, this makes a big difference if you are familiar with testing of water for E. coli. In the case of E. coli, you get 100 milliliters or perhaps 1 liter. In the case of parasites, because there's no enrichment method, we need to collect large volumes of agricultural water to be tested for the presence of these parasites. We have many challenges. One of them is in the lab, that we don't have any animal model we can grow it in the lab. Therefore, the availability of this parasite to do studies is very limited. We need to find simple and fast detection methods to test it in environmental samples and food samples, and more important, something that is not expensive, that it will allow the farmers to test and use it more often. We all are looking for methods to do fingerprinting, genotyping tools. It works. There are methods that work very well with clinical samples. And again, if a person has cyclosporiasis, there's a large number of oocysts in the stool samples. But when we go into the world of the environment and the world of foods, the contamination can be 10 oocysts, 20 oocysts, and these tools, the genotyping tools, are not there yet for us to be able to actually associate one particular illness with a particular food commodity using DNA tests. Critical for us, for Cyclospora, is prevention, because, again, we don't have chemical methods to inactivate this parasite. Therefore, we need to prevent the introduction of this pathogen in the field. There's a lot of interest by the food industry to identify the sanitizers, identify a method that can be easily used, that is cost effective, and again, that is effective to cure this organism. The outbreak investigation is getting faster and faster every year. And again, the questionnaires now are online, so the testing, the interviewing of the individuals is faster. The testing, as I mentioned to you, using culture independent tests can be done very fast, and therefore we are detecting more cases. So from this perspective, detecting public health labs, it's working very well, as well as epidemiologists. We still need to work a little more when it comes to the testing of certain food commodities and water on time in order to be able to stop this outbreak. That's all I have to tell you today. Thank you very much. And again, if you have any questions for me, I will be glad to answer them. Thank you.